Hello everyone, this is Jason from STEM Nerds. In today's video, we're going to be talking about the cell walls of bacteria, of prokaryotic organisms, because it is very interesting. In the previous video, we talked about all the cell structures and organelles that I think are important. You can watch that video. I'll put it in the upper right-hand corner as well as in the description below. But here we focus specifically on one thing, and that is the cell wall of bacteria, because it is hugely important when somebody has a bacterial infection. One of the first things a healthcare professional wants to know is, what does the cell wall structure look like? And we have two basic designations. They're either gram-negative or gram-positive. And it really is based on the way these cells will stain using a staining process called the gram stain. And so once you know if it's a gram-negative or gram-positive cell wall structure, it gives you a much clearer indication on how to treat that infection. So let's go take a closer look at these structures. So just as a review, this was a prokaryotic bacterial cell, and we went over this in the previous video. We talked about all the parts and pieces, and then I said, hey, that cell wall that's labeled over there on the left-hand side, that is a really unique structure, and we need to dive into that a little bit deeper, and so that's what we're going to do now. So this is a gram-positive cell wall structure. Now, there's a few things that we need to point out. I'm going to show you a picture in a second here, and it's going to all be labeled. But right here, you can see the gold. You have a phospholipid bilayer, something we've talked about before. So we've got this amphipathic qualities of the phospholipid bilayer, where the cell membrane has the charged heads and then the hydrophobic tails. And then we can see here we've got a whole bunch of stuff outside of that, and that is the actual cell wall exterior to the membrane itself. Then we've got some things sticking up. And so let's go ahead. I'm going to show you a picture of the label. And so if you're looking at this right here, below the phospholipid bilayer, so at the bottom of the picture, that would be the cytoplasm, and all that purple stuff and all those circles and everything, that would actually be the exterior of the cell or the environment. All right, so this is a drawing of a gram-positive cell wall structure. And again, to orientate ourselves, the bottom of the picture, that would be the cytoplasm. The top of the picture would be the actual exterior environment of the cell. And we can see here we've got a phospholipid bilayer. That's our lipids. We can see that there are proteins. The proteins can either be peripheral or they can go all the way through the membrane. But the really important part exterior to that is we've got this layer, once again, of peptidoglycan. I know it's a big fancy word. Sometimes we just initial it PTG, but peptidoglycan is all of that blue stuff that you see outside there. Now, with the gram-positive cell wall structure, what we need to know is that there are often many layers of peptidoglycan, so the cell wall is going to be very, very thick. We're also going to see that we have these structures here, and they're different colors and different shapes, and that's because they are different sugar structures. One of them is called NAG, N-A-G, and one of them is called NAM, N-A-M. I'm going to fill in some details there in a moment. The other important thing that you're going to see is a structure called teichoic acid. So teichoic acid is a structure that kind of holds all of those different layers up, kind of like the support pillars of a high-rise, of a building. And so that teichoic acid is really important. We generally only find that in gram-positive cells. And again, if we're differentiating between gram-positive and gram-negative, the gram-positive have many, many layers of peptidoglycan along with that teichoic acid. And so here's basically what that NAG and NAM would look like from, I guess it would be a top view. And so NAG stands for N-acetylglucosamine, NAM, N-acetylmuramic acid. Again, for my students, it's just NAG and NAM, but you can see that they're alternating. And the other thing I want to point out here in this picture is you're going to see this thing called the tetrapeptide bridge. It's just labeled tetrapeptide here, or the peptide link. Together, we call that the tetrapeptide bridge, and that is what is holding the NAGs and NAMs together, kind of like a single blanket. It's like the stitching in a blanket, so it's holding everything together across a single layer, where the teichoic acid previously was holding up all the different layers. All right, so now this is our gram-negative cell wall structure. You're going to see it looks way different from gram-positive. It starts out the same, however. The lower phospholipid bilayer is still there. We see that light blue area there, that's a protein. So we have a phospholipid bilayer. And then all the little purple circles in the middle, that is our peptidoglycan. But importantly and very obviously, we have a second membrane, what we call an outer membrane. And so gram-negative cell walls, gram-negative organisms, have two membranes. They have an inner membrane that's closest to the cytoplasm, that would be towards the bottom of the picture. And they have an outer membrane, which is closer to the top. The peptidoglycan is usually just one to maybe just a couple layers thick, so it's not nearly as thick as what we see in gram-positive. So just like before, I'm going to show you a labeled picture so that we know all the parts and pieces that are important for a gram-negative cell wall structure. All right, so here we go. Again, we're looking at basically the side of the gram-negative cell wall towards the bottom. You've got the cytoplasm out where those things are sticking up. That would be the external environment of the cell. So let's work our way from the bottom towards the top. So at the very, very bottom, we see here on the right-hand side, it's labeled cytoplasmic membrane. So it's our phospholipid bilayer. You can see some proteins are embedded within that as well. Now, in the center between the two membranes on the left-hand side, we're going to see this periplasmic space, otherwise known as the periplasm. This is the location, again, that's between the two membranes, the inner and outer membrane, and it's also the area where the peptidoglycan is going to exist, and it's going to alternate NAG and NAM just like the gram-positives did. But gone is the teichoic acid because we don't have all these layers that we need to support. Now, where things get interesting is in this outer membrane because we actually have two different structures. Okay, now you got to follow me here. On the inner leaflet of the outer membrane, you're going to see it's labeled on the right-hand side phospholipid. So those phospholipids are basically the same phospholipids that we see in this inner membrane. But if you look at the top, that outer layer of the outer membrane is called lipid A. And so it's a different type of lipid structure. And that's really important. In fact, that whole thing, we call that LPS, lipopolysaccharide. And that consists of a lipo is a lipid. Polysaccharide, these are sugars that are sticking up from the top of this gram-negative outer membrane. And this LPS stuff is really, really nasty. It can absolutely cause all kinds of problems in the human body, and our immune system generally freaks out in the presence when LPS gets in the body. So this is a really dangerous thing to have in high concentrations. Also, if you have different types of a certain type of bacteria, like let's say E. coli, maybe you've heard E. coli and it's got a bunch of letters and numbers after it. What those letters and numbers are designating, what they mean, are the different types of sugars that are associated with that LPS, with that lipopolysaccharide. All right, so this is LPS, lipopolysaccharide, and it's kind of turned on its side. So on the right-hand side, you're going to see the lipids. That would be moving towards the center of the outer membrane. And then you've got that core polysaccharide. And then beyond that, you've got that O-specific side chain. That is what is going to be sticking up into basically the environment of the surrounding environment of the cell itself. Now, I explained this process in a different video. Really, this is a lab procedure, and so when I do the lab videos, I explain it there. But these are the results of gram-negative and gram-positive stains. So on the left-hand side, where you've got the pink, that is actually the result of a gram-negative organism. On the right-hand side, the purple is gram-positive. And so once again, this is so important because the diagnostics on the medical side and how you treat gram-negative microbial infections relative to gram-positive are very different. They require different antibiotics. And if you get that part wrong, you're going to have all kinds of issues. And the gram-negatives are really, really dangerous. Like I said, especially when they get into the blood, they can cause toxic shock and all kinds of things. Not that the gram-positives are safe. They're not. But the gram-negatives are really, really important. All right, so I just wanted to spend a few minutes talk about the differences between gram-positive and gram-negative cell wall structures. As always, thank you so much for being here. We'll see you next week.